Well, good afternoon, everybody. It's a real pleasure to be here. Nice to see many friends in the audience in person after such a long time. My name is Suyash Prasad. I'm the Chief Medical Officer and Head of R&D at Taysha Gene Therapies, and I'm looking forward to telling you a little bit about Taysha over the next 15 minutes or so. Of course, I will be making some forward-looking statements. This is our legal disclosure. Taysha is a fairly new company. It's been around for about 18 months. We did a Series A in March last year, a Series B in July, then took the company public in September. So we've been public for about a year, and our focus is on rare and severe neurological diseases, principally affecting children, also adults, and also deeper in our pipeline, some of the commoner neurological diseases. Taysha by the numbers, so a few highlights here. We have a really world-class partnership with the University of Texas Southwestern in Dallas, which is really fuels our pipeline, which is extensive. We have 27 programs in development. We have one program which has some is in pivotal clinical stage, and I'm going to share some of the data from that particular program as this presentation progresses. By the end of this year, we intend to have 5 INDs or CTAs open, and across our portfolio of patients, we have an addressable population of over 500,000. This is the pipeline. I'm sorry, it's very small. You can't quite read it, but what I will emphasize is the fact that our portfolio of neurological diseases is split into 3 franchises. The neurodegenerative franchise, which is looking at diseases where there's an ongoing progressive loss of neuronal tissue. The neurodevelopmental diseases such as Rett or Angelman syndrome, where there is disordered neurology, and we feel these diseases are somewhat amenable, quite highly likely to be amenable to reversibility, and the genetic epilepsies, which are increasingly being described, and the molecular underpinnings of such diseases are increasingly being elucidated. Now, we have a large portfolio, as you can see, 27 programs, 5 INDs by the end of this year. And part of the reason for our ambition and our ability to move things forward is the fact that we take a platform-based approach to developing gene therapies. So certain things are common from program to program. Everything we do is AAV9. It's a well-known serotype. It's the serotype that has most experience in clinical practice. Everything we do is HEK293 mammalian cell suspension manufactured. Also, there's the most clinical precedent for this manufacturing process. And everything we do is delivered intrathecally. So we give drug intrathecally into the base of the spine to allow it to perfuse the brain on the spinal cord. It's something I'm comfortable with. I'm a pediatric intensivist by training, and I've actually given a lot of intrathecal drug in my career. Not gene therapy, of course, but oncology and anesthetic drug. But it does give you good delivery to the brain and spinal cord. And you'll see from our data in our lead program that it works very effectively. That's what's common from program to program. Where the creativity arises in what we do is what goes actually in the capsid. So we have a number of programs that just simply replace the gene, the full-length gene. We have a number of programs where the gene is replaced but also regulated with a strip of microRNA, for example, or microRNA binding sites that help regulate the degree of expression, which is important in a disease such as Rett syndrome, where there's a risk of overexpression toxicity. We vectorize RNA, so we include short hairpin RNAs within our capsids to down-regulate or knock down the protein. And also, as you all are aware, sometimes the gene's just too big to fit into a capsid, so we're thinking about mini-gene approaches as well. How do we truncate a gene, make it smaller, such that it can actually fit into the capsid, yet still retain its functionality? A lot of our creativity in this sphere really comes from our partnership, our collaboration with University of Texas Southwestern, led by Burj Manassian, who's the Division Chief of Pediatric Neurology, and Stephen Gray, who is the head of the Vector Core. Stephen is a very renowned, fine gene therapy scientist, well-published, really very patient-focused, and really wants to move his academic research forward into the clinical situation. And the partnership with UT Southwestern works very, very well. Burj and Steve lead this organization. There's about 8 to 10 principal investigators or clinical investigators below them, and a group of about 60 or 70 working as part of the gene therapy core. And this group really functions as our discovery group, and everything that, all the programs that we move forward into IND enabling preclinical studies and into clinic really come from UT Southwestern. It's a really wonderful partnership. So let me tell you about our lead program, giant axonal neuropathy. This is the first ever dosed intrathecal gene therapy. The first patient dosed was in 2015 at the NIH. Giant axonal neuropathy is a neurodegenerative disease. The underlying pathology is a mutation in the protein gigaxonin, which is involved in metabolizing, breaking down waste, old structural protein within the axon. In the absence of gigaxonin, you get this swelling of the axon, which you can see in the image on the right-hand side. And what that does is it impairs electrochemical transmission down the actual axon, which results in the clinical symptomatology of giant axonal neuropathy. In terms of the natural history of the disease, in the yellow you've got the early onset form of GAN. In the blue you've got the later onset form of GAN, which is often misdiagnosed as Charcot-Marie tooth. And when we look at the numbers and you model from the publications in epidemiology, it appears there's probably about 2,400 patients with GAN mutations around the world that will be amenable to the gene therapy approach. So definitely a rare disease, but still a significant opportunity. Now, how does the disease present itself? Well, children are born and they look fine at birth. Around the age of 2, they start to develop this wide-based gait and a bit of unsteadiness on their feet. This is because they lose sensation to the feet. They start to stumble, they start to fall. There's a progressive motor decline, bulbar function goes, vision goes, and these children usually end up in wheelchairs around the age of 10, on a ventilator around the age of 15, and then usually they die in the late teens or early 20s. So it is a relentlessly progressive neurodegenerative disease with no treatment options whatsoever. This is the construct. You've got the full-length human gigaxonin gene wrapped up in an AV9 capsid. We use a jet promoter, which is a small synthetic promoter, which is moderate in strength but ubiquitous. And we had a very extensive preclinical package supporting this, so animal studies in mice, in rats, and in NHPs. And I want to just pause for a moment and tell you about one of the findings in our preclinical studies. One of the big topics in the world of gene therapy at the moment is safety. One of the specific topics in safety is this notion of DRG toxicity, dorsal root ganglion toxicity. I personally don't like the phrase toxicity because it's really only been found as a histopathological finding in NHPs with very little actual functional or no functional compromise. And I think it's important to realize that some diseases actually have dorsal root ganglion pathology as part of the disease. So this idea of avoiding the DRGs to prevent toxicity is not appropriate for every disease. And giant axonal neuropathy is one of those diseases. And you can see here in the diagram in the middle, this is the GAN knockout mouse, the dorsal root ganglion of those mice. And what you see is protein accumulation and associated degeneration. And in the diagram on the right-hand side, you actually see a treated GAN knockout mouse pathology slide with the protein inclusions being reduced and regeneration of the tissue. So you're actually showing improvement of the DRG in this particular disease. And I think it's important to remember that when we're thinking about adverse events in the world of gene therapy. Sometimes you actually need to, in this particular case, get drug into the DRGs to affect a histopathological improvement and thereby a clinical improvement. This is the clinical trial design. As I say, it started dosing in 2012. There's 4 doses, a low dose of 3.5 E13 total VG, the medium low, the medium high, and the high dose of 3.5 E14 VG. I'm going to share all the doses apart from the high dose that's yet to come later this year. We give the drug intrathecally. We cover with prednisolone and rapamycin as immunosuppressives. It's important to remember that these doses are total VG and not VG per kilo, which means that you're actually giving a high dose of drug targeted to the brain and the spinal cord, but actually a relatively low dose of drug in comparison to systemically administered gene therapies, which once again adds to a favorable safety profile. This is the natural history study that was started 2 years prior to the interventional trial, and what you see here is a reduction in the key primary endpoint, the MFM32, which is a neurological score. you see a reduction of about 8 points per year. A 4-point decline in the MFM32 is recognized as being clinically meaningful. So there's a clinically meaningful change in this MFM32 score over time, regardless of the age or the level of functioning of the child. So you could be a 5-year-old with a score of 80 or a 10-year-old with a score of 60, and you still drop by about 8 points per year. That's the natural history. Now, this is the treatment with the medium low and the medium high dose. What you see here is that you have a natural history. I'm going to try and show you on the pen here. You have the natural history slope, as we've already talked about, 8 points per year, and with the medium low and the medium high dose, blue is the medium low dose, green is the medium high dose, you see a stabilization of disease progression. So this translates to an 8-point change in the clinical trajectory of patients who have been treated. That's over the course of a year. If you look at every patient individually, once again, in the green you've got the medium high dose, in the blue you've got the medium low dose, you actually see a stabilization once again. And in fact, if you look at the lowest performing patient with a medium low dose, they drop by about 20 points, 22, 23 points prior to treatment. The vertical lines where they get treated, and they stabilize out. And what you see here is that you've got long-term safety, long-term durability, and long-term efficacy over the course of 3 years of observation. So it's really definitely a maintained effect. We performed Basin analysis just to add some sensitivity and greater clarity. What you may understand from a Bayesian analysis, there's 3 things to remember. The first thing is it's been around for a lot longer than standard frequentist analyses. Increasingly is being accepted by regulators, especially for these small patient populations, and especially for these rare diseases where you get a big change in clinical endpoint, and you end up with a probability value. And it's a little small to read, but what you can see here is that the probability of the medium high dose of a clinically meaningful effect is 98.3%. The probability of any effect of the medium high dose is 99.9%. So really quite dramatic improvements as assessed by the Bayesian approach. A second endpoint I wanted to share is visual acuity. This is really important from a humanistic quality of life perspective. It's the one clinical feature that parents and families really want to preserve, because it's the last way of communicating with their child as they're losing speech capability, movement. They really want the child to be able to see so they can pick up on visual cues. What you see here with the visual acuity scale, the logMAR, and to orientate yourselves, a logMAR score of 0 is normal, 0.3 is mild visual impairment, 0.7 is significant visual impairment, 1.2 is blindness. You can see that a logMAR scale is going up for every patient apart from one outlier, and then they stabilize out on treatment. So once again, we're showing stabilization of disease. Safety has been very good. On the immunosuppressive regime of prednisolone and rapamycin, the dog's been well tolerated. No dose-limiting toxicities have been found. No transaminitis, no evidence of thrombocytopenia or MRI changes. So very comfortable with the safety profile here. And in terms of next steps for this program, you can see we're very excited about this data. We show clear stabilization of disease progression at the medium low and the medium high dose. The high dose is yet to come. We show dose response, we show long-term safety, long-term durability, long-term efficacy, and we have a great natural history study. So we're looking forward to having discussions with regulators later this year and early into next year, FDA, EMA, MHRA, to map out a pathway towards clinical approval. So we've got lots happening at Taysha. We've got a lot of value-generating catalysts coming up over the course of the year. High-dose data from GAN, biomarker data from our GM2 gangliosidosis program. We'll be dosing the first patient with CLN1 later this year, and additional clinical data from the CLN7 program. We're building out our manufacturing facility. We've got a 200,000 square foot facility in North Carolina that should be up and running, producing GMP material 2023. And then we have another 5 programs in IND-enabled preclinical work, which we will be discussing during the course of next year, hopefully submitting INDs for those programs next year. And you hear about the first one of those at our Angelman Investor Day later on this year. On that note, I'd like to thank you for your attention. Very happy to be here once again, and I think we're running out of time, so I'm not sure if we have time for questions or I can come and take questions at the end if you come and find me. Thank you.